Bismillahirrahmanirrahim. Paper C Genetics. Topic Gene mutation. When a change occurs in a gene, it is known as the gene mutation. Mutation is the sudden heritable change. Mutation normally occurs during the process of development and it is frequent at the time of gamete formation, during zygote division, or in the vegetative tissues. A gene is composed of protein and DNA. Protein consists of different types of amino acids. Any change in the composition or the position of amino acid or any replacement of the amino acid cause mutation. Mutation occur frequently in nature in many organisms. For example, mice, pigs, Drosophila, man and rabbit. Types of mutation. Mutation can be classified on the basis of 4 reasons. First one is the according to the type of the cell. According to the type of the cell, mutation are of 2 types, somatic and gametic. First one is the somatic mutation. When the mutation occurs in somatic cell, the non-reproductive cell of the body, it is called somatic mutation. This type of mutation, it takes place in one cell or its daughter cell and it cannot be inherited. Therefore, it is not very important. Second type is the gametic mutation. When a mutation occurs in a reproductive or sex or germ cell, for example sperm and eggs, this type of mutation is known as gametic mutation. This type of mutation can be inherited from one generation to the next. Therefore, it is genetically very important and significant. It may cause evolution. Second type, according to the size and quality. It may be of 2 types, point mutation or gross mutation. First one is the point mutation. When mutation occurs in a small part of a DNA molecule, either in a single nucleotide or a pair of a nucleotide, it is known as point mutation. Point mutation have further types: deletion, addition, substitution. First one is the deletion point mutation. When mutation occurs due to loss of some portion in a triplet codon of a gene, this type of mutation is known as deletion. For example, a triplet codon made up of triple U, if one nucleotide is deleted or removed, results in the deletion point mutation. Second is known as the addition point mutation or insertion point mutation. When there is addition of one or more extra nucleotide to a gene, it is known as the addition or insertion point mutation. A triplet codon made up of 3 nucleotides, if an extra nucleotide is added, it results in the addition point mutation. You can see in the diagram the chromosome before the mutation. When a part of it is deleted or removed, this is the condition of the chromosome after deletion point mutation. Second, duplication, the condition of a chromosome before mutation. When a similar part is added, so this is the condition of the chromosome after mutation, that results in duplication point mutation. Third is the substitution point mutation. When a nucleotide of a triplet is replaced by another nucleotide, it is called substitution point mutation. The example of the substitution mutation is sickle cell anemia. The normal RBC is flexible and disc shaped, but in the case of sickle cell anemia, the red blood cell, they become more stiff and curved in the shape of an old farm tool that is known as sickle. Because of this shape of the red blood cell, the disease is known as sickle cell anemia. In the case of normal RBC, the triplet codon present on the messenger RNA is GAA, responsible for the amino acid glutamic acid. In the case of sickle cell anemia, the codon present on the messenger RNA is GUU, responsible for the amino acid valine. So if glutamic acid is replaced by means of valine, it results in the disease known as sickle cell anemia. So this is the example of the substitution point mutation in which a triplet codon is replaced by another triplet codon. Second type of mutation is the Gros mutation. When more than one nucleotide pair or entire gene are affected, that mutation is known as the Gros mutation. In the case of Gros mutation, gene locus may be changed. It takes place by two processes. First one is the translocation. Second one is known as the inversion. In the case of translocation, there is the movement of gene takes place to a non-homologous chromosome. The chromosome segment and their genes change position. So an intrachromosomal translocation, a segment breaks off the chromosome and rejoin it at a different location. You can see in this diagram translocation, Gros mutation, two chromosome, these two parts are translocated, as a result of which after the translocation the condition of chromosome. Second type of Gros mutation is inversion. The movement of gene takes place in same chromosome. This is the diagram of the inversion Gros mutation. Before the mutation, the genes present on the chromosome, their sequence is A, B, C, D and E. But after the mutation, the sequence became A, B, E, D and C. So the two genes E and C their place is inverted, results in the mutation known as inversion. Third type of the mutation, according to its origin. The origin of the mutation may be of two types, spontaneous and induced. Spontaneous mutation, the mutation that occurs suddenly in nature, also known as background mutation. Second, induced mutation. This is the artificial type of the mutation. It is induced artificially in the bodies of living organisms. The causes of mutation is radiation or change in the temperature or any other factor like UV rays. According to the phenotypic effect, the mutation may be of following type. First one is the dominant mutation. When the mutation appears physically or phenotypically, it is known as dominant mutation. For example, in man, a disease is called aniridia, in which the iris of the eye is not present. It is the example of the dominant mutation. Second, recessive mutation. When the mutation does not appear physically, it is called recessive mutation, but it can appear in the next generation. For example, color blindness. And last type of the mutation according to the phenotypic effect is the lethal mutation, the type of mutation that cause the death of the person. So it is very dangerous. For example, sickle cell anemia. Now the mutagens. The substances or agent that cause the mutation is known as mutagen. The mutagens may be of following types: ionizing radiation, ultraviolet radiation and chemicals. First one is the ionizing radiation. It may be X-ray, gamma ray that cause damage to the DNA. When these rays enter the cell, they are absorbed in the atoms. Their energy is transferred to the electron of the outer shell. Due to this energy, electrons are released from the outer shell. By the releasing of electron, atom become ionized, contain electron without pair. Here it is electron. Atom of the cell are present in the water. They cause damage to the DNA. The free radical means that contain electron without any pair. So the free radical, it breaks the phosphodiester bond of the DNA, causing DNA damage. Second type of the mutagen, ultraviolet radiation. These are the parts of sunlight that cause sunburn. These rays contain less energy as compared to the X-rays, so therefore they do not produce free radicals. Third type of the mutagen, chemical mutagen that cause damage to the DNA. Three different types: chemicals that are like the DNA nucleotide and may be wrongly attached to the DNA. Second, they remove the amino group from the nitrogenous bases, causing damage to the nitrogenous bases. Third, they attach hydrocarbon to the nitrogenous base and damage the structure of the nitrogenous base. Modern industrial societies are producing and releasing many harmful chemicals in the environment which cause damage to the DNA. These chemicals are known as mutagens.